Okay, hi everybody. So this is the single gene disorders lecture from the human genetics module, which is a year one module on the biomedical sciences degree at Bradford University. So the aim of today's lecture is to enable you to develop a bit of more of a critical understanding about the way that variation in the genome contributes to disease. So you should have had a lecture now, hopefully you remember my lecture on genetic variation, where we discussed why it's important that mutation and genetic variation exists in order to be able to drive forward evolution and allow organisms and populations to adapt to environmental pressures and changes in circumstance. We also discussed the types of genetic variation that exist, the types of mutations which exist, etc. What I want to do today is give you some examples of how those particular changes in DNA sequence, those mutations, lead to disease, and the different ways in which these mutations can result in disease, and the different ways in which these genetic diseases can be influenced themselves by external factors. So by the end of the lecture, hopefully you can discuss the role of single gene mutations in the development of a range of human diseases. So we're going to start by addressing the question of how genes actually cause disease. So, and then lead on to discussing some of the issues around prevalence of single gene disorders. As you'll see, single gene disorders are very rare, but they're not so rare that they're not significant with regards to the number of live births that are affected. As I mentioned on the last slide, there's numerous different factors that can actually affect the phenotype of single gene disorders. So it's not always just a case of a particular mutation in the gene directly leading to a phenotype. The mutation which affects the gene can lead to different phenotypes depending upon external factors, often. And we'll discuss what those external factors are and the different types, or if you like, of single gene diseases that exist in humans. I'm going to give you some information about mutation nomenclature, so it's important that you understand how scientists describe the different mutations which exist in DNA. Certainly if you go on to have a career where you work in the NHS genetics service, which is an ever expanding arm of the NHS, it's going to be important that you have a clear grasp of how mutations are described in the literature. And then we'll finish by summarising the main points of the lecture. So let's start then by considering the four main ways in which genes can actually give rise to human disease. So the topic of this lecture is single gene disorders. So we'll start with this. So in single gene disorders, the mutation in that particular gene is more or less the sole cause of the symptoms. So there are some exceptions, but in the majority of cases it's the mutation in a gene which is having a direct effect on the gene product, the protein, and that effect on the protein is directly then related to the actual outcome for the individual, the actual symptoms, the phenotype that individual displays. So we're going to be focusing on these types of mutations in this lecture. In the next lecture, we're going to talk about multifactorial disease, sometimes called complex genetic disease. And this is a lot more obtuse to unpick and difficult to map out, if you like, with regards to the effect that mutation and variation is having on these particular types of human disease. So this is where genetic variation interacts with the environment, a wide variety of other factors, in order to lead to a particular illness. And really it's these complex genetic diseases which if we had an option, we'd really like to be able to understand better and to hopefully treat and ultimately cure, because it's the complex genetic diseases which encompass the big killers, if you like, of humans in healthcare, which are heart disease, cancer, those sorts of illnesses. I'm going to talk more about those in the next lecture. So in addition to single gene disorders and complex gene disorders, you can also get illnesses and syndromes and diseases arising due to chromosomal effects. So aberrant chromosome numbers or abnormal chromosome structure are both able to give rise to human disease. So again, Down syndrome, trisomy chromosome 21, Turner syndrome, which is the lack of a second X chromosome. Both of those are examples of aneuploidy, of changes in chromosome number leading to disease. We gave a good example of aberrant chromosome structure in the last lecture for CML, chronic myeloid leukemia, whereby the BCR gene is translocated in such a way that it becomes fused to the ABL gene to create the BCR-ABL oncogene. And this gene gives rise then to this type of leukemia. And finally, there are also mutations which can occur in the genome of mitochondria. So I would hope that when you had your tutorial with your tutor, they explained that mitochondria in eukaryotes possess their own genome, and this itself can be mutated occasionally in individuals to give rise to human disorders. There are several really nasty types of diseases that arise due to mutations in mitochondrial DNA. One example is the syndrome MELAS, which stands for, if I remember rightly, mitochondrial encephaly and acidosis with stroke-like syndromes. It's a bit of a mouthful, that may be wrong, but in essence it's a nasty disorder which manifests itself as just random unexpected stroking in the individual, and that arises from a mutation in the mitochondrial genome, which is obviously X chromosomal. It's nothing to do with the genome in the nucleus. So they're the four major ways actually in which genetic variation, in which mutations in our genes can give rise to disease. So the first thing to emphasise, and I kind of touched on this in the last slide, is that single gene disorders are very rare events. So we're talking about diseases here which are often incredibly rare, affect only one in several tens of thousands, sometimes several hundreds of thousands of people. In fact, there are single gene disorders which are so rare that we only have a handful of examples of people suffering from them in history. Sometimes there are actually no people alive who suffer from this documented disease, or certainly there are only 3 or 4 people on the planet who suffer from it. So these are very rare, specific genetic disorders. And they kind of, again, fit into this idea that we talked about in the last lecture, whereby everybody has unique mutations present, which haven't been witnessed before or are very, very, very rare. And it's these unique mutations, if you're extremely unlucky, that can give rise to these very unusual, very rare disorders, which are being reported all the time, new examples of mutations in genes which we've just not seen previously. Having said how rare these things are, they still give rise to a significant number of live births who possess and suffer from these genetic single gene disorders. So from a healthcare point of view, even though they're extremely rare diseases, they are significant and they are worthy of research, and they're very important in global healthcare. So let's have a look at the frequency then of some of the most common genetic diseases, and this is per 1,000 live births. Well, by far the most frequent are congenital malformations, so cleft palate, things like that, and around 20 live births out of every 1,000, or 1 in 50 live births will actually have some form of congenital malformation. So one of the things really that strikes home, I think, when you start talking about these single gene disorders as a parent, I've got a young girl and a new son who's not a year old yet, is that when, as an academic and as a student, when I learned about these single gene disorders, the frequencies of occurrence have seemed very low, and to so low that they're not really a concern. But I can assure you, if you're a parent, if your partner's pregnant and you're expecting a child, then you start to look at the information that you're given by the NHS relating to Down syndrome or relating to cystic fibrosis, and those odds don't seem so long anymore. So that kind of is, I guess, a better way of emphasising the importance of these diseases, even though they're very rare. So the second most prevalent type of genetic disorder then is Down syndrome. So around 7 births out of every 1,000 births will result in a a child that has got Down syndrome or a similar chromosomal disorder. So that's not, we're talking about 1 in every 250, one in about every 150, live births there. So, again, 1 in 150, seems you wouldn't want to, you wouldn't want to put money down at a bookies perhaps, but those odds are not that long if your partner's pregnant and you're looking at having a child. And obviously one of the issues with Down syndrome is that the likelihood of the child having Down syndrome increases quite drastically with maternal age. So particularly once the mother gets over 35 years old, if you look at a curve of incidence of Down syndrome, you'll see that curve starts to rise steeply after 35. And that's actually due to a process called homologous non-disjunction, which is a process that can occur during actual nuclear division, whereby an aberrant number of chromosome 21 gets dragged into the resultant daughter cells in such a way that you end up with one cell that doesn't have any chromosome 21s and one cell that has 3. And this chromosomal non-disjunction is thought to be linked actually to, if you recall back to the cell cycle lecture, the way that chromosomes are held together by cohesin at the metaphase plate. And it's a fact that the chromosomes in the egg cells of women are actually held at a state of metaphase, at mitosis, for many years prior to division. And it's thought that as the age of the woman increases, the actual likelihood that the machinery responsible for maintaining that chromosome state starts to deteriorate in such a way that the likelihood then of chromosomal non-disjunction occurring in those cells and an aberrant number of chromosomes being inherited increases significantly as the woman enters more mature age, approaches 40 plus. It's a little bit more complex than that, but that's the gist of it. So that's Down syndrome. We get on then to the more rare, if you like, disorders. So we have the autosomal dominant, autosomal recessive and X-linked disorders. So these are single gene disorders now which just affect individual genes within the genome. X-linked disorders, about 1 in 500 live births will suffer from an X-linked disorder. Haemophilia is by far the most common example of that. Autosomal recessive disorders, again, probably the most common, cystic fibrosis, particularly in Caucasian populations, where 1 in 500 live births, again, will result in a child which has cystic fibrosis. And remember, this is a disease that has no cure. Treatment has improved the prognosis of cystic fibrosis patients, but if your child is diagnosed with it and is born with cystic fibrosis, they have a lifespan of 35-40 years with current medical treatment. Sometimes less, sometimes maybe a little more. And finally, we have autosomal dominant disorders. By far the nastiest example of these is Huntington's disease. Huntington's disease is particularly nasty because of the genetics of its inheritance. So we've talked a little bit about this on the genetic variation lecture, but in a nutshell, Huntington's disease is an autosomal dominant disorder. So you only need one copy to be inherited in order to inherit the disease. And the nasty thing about the Huntington's disease is that it doesn't present actually as a disease until quite late in life, until around 35-40 years of age. So obviously by that point in life, most people have already made the decision of whether they're going to start a family or not, and may have had children of their own. And what happened when geneticists kind of uncovered the gene that was responsible for Huntington's Huntington's disease, and identified it as a single gene disorder, was that there were numerous cases of this disease which then could be tracked through family trees by genetic counsellors and geneticists. And what this resulted in really was a situation where you had perhaps elderly people in their 60s who were dying of Huntington's disease, or had died of a neurodegenerative disease which was subsequently confirmed as Huntington's, who'd already had children of their own, who were perhaps in their early or mid or late 30s. So at the point where the gene was identified, these individuals then would be offered the opportunity to be genetically tested for the disease, whether they had the disease or not. And that's a massive decision for that person, because remember, this is a disease with no cure. So if you're 30 years old and your parents died of a neurodegenerative disorder which is suspected to be Huntington's, you've got a big decision on your hands whether or not you want to be tested or not. If you decide not to be tested, Are you then just waiting for symptoms to appear, living in fear of the disorder? If you are tested and you're positive, how then do you proceed with your life? You confirm that you are going to get ill and die from this neurodegenerative disease in the next 10 to 15 years. So it can have a huge impact on people's lives. But it even goes further than that, because those individuals who are 30, 35, 40 may have already had young children of their own who maybe were very young. Do those children get tested? Do you want a child who's 10-year-old to know that they have inherited this disease gene which is going to kill them when they're 45, 50-year-old? Is that something you want your child to live with? Would you rather they didn't know? And more recently, a different dilemma has arisen where families have had a situation where the original children of an elderly person who's died, so the people who are like maybe in their 40s, late 40s, don't want to be tested. They're asymptomatic yet, but they have a child of their own who's 18 and who can decide that they do want to be tested. They have the right, obviously, to be genetically tested for the disorder. Now, if that child obviously is tested and they turn out to be a carrier of the mutated allele, then that automatically diagnoses their parent, irrespective of whether that parent wanted the test or not. So this is a really delicate area for genetic counselling and for geneticists, Huntington's disease, and a really terrible disease that really tears through and destroys entire family trees. Autosomal dominant disorders, relatively rare, predominantly because of achondroplasia and other things like that, we'll talk about in a second. Now, one of the interesting things then about single genes disorders and the diseases which arise from single gene disorders is this. As things stand, there are around 10,000 diseases that have been attributed to mutations in single genes. So in other words, there are about 10,000 different diseases that we're aware of, which we know now result because the results of a mutation in a single gene. So what that tells us is that there must be more. What that tells us is that different mutations within the same gene must be able to give rise to different diseases. Yeah. So there are only 6,000 genes in which mutations have been identified, but there are 10,000 diseases which we know arise from mutations in single genes. So therefore there must be single genes which can be mutated in different ways to give rise to different diseases. And this is the phenomenon which is known as genetic heterogeneity. So the concept that different mutations in the same gene can give rise to different diseases. And as I mentioned before, anything in red, that's a definition. Make sure you understand what the definition of those words are, because it's the sort of thing you need to understand for the assessments. So an example of genetic heterogeneity then. Well, a nice example is a mutation in the gene that encodes for the fibroblast growth factor receptor 3 protein, FGFR3. So this is a picture of the FGFR3 protein. You don't need to learn this. This just is quite a complex example of how the protein works. Effectively, it's a membrane-bound signalling protein, FGF receptor 3. How it works is the FGF substrate, if you like, the fibroblast growth factor protein binds to the receptor on the exterior of the cell, and that causes changes to the interior of this FGF receptor 3 protein, which then in turn trigger signalling cascades inside the cell and lead to different types of genes being switched on and being switched off. So it's this kind of signalling cascade that we've talked about previously when we've talked about the cell cycle and growth factors triggering changes in gene transcription. Anyway, it's not that the molecular biology of it isn't what I want you to understand from this. What I want you to understand from this is the fact that FGF receptor 3 is a good example of genetic heterogeneity. So for example, if you mutate the FGF receptor 3 protein in such a way that the proline residue, which is normally located at the 250th amino acid of the protein, is altered to an arginine residue, what you generate is a protein that gives rise to a syndrome called Muenck syndrome. So this is an autosomal dominant syndrome that's characterised by premature and aberrant fusion of the cranial bones in the head, in the skull. Yeah. Now, exact same gene, different mutation. If you mutate the glycine residue, which is present at 308 amino acids of the FGF receptor 3 protein to an arginine, what you actually do there is you generate a syndrome called achondroplasia or dwarfism. So this is the mutation that Weeman has, I believe. He has achondroplasia. And the interesting thing is it's exactly the same protein, slightly different point in the protein is mutated, very different disease outcome. And it's this sort of thing that gives rise to the phenomenon of genetic heterogeneity. So if you look on here, this can be explained by the fact that one of these mutations affects a particular region of the protein outside of the cell, whereas the other mutation affects the region of the protein inside the cell. And so altering the protein structure outside or inside the cell is going to alter in a different way how that receptor is functioning, and that different alteration is what gives rise to the different disease. So in some ways, genetic heterogeneity kind of reflects the multifunctional nature of proteins. There are more examples of genetic heterogeneity because it's also genetic heterogeneity. Sorry, I get tongue-tied. Genetic heterogeneity can also be used to explain the phenomenon whereby mutations in different genes can lead to the same disease. So we've just looked at an example where you can mutate a gene in two different ways and get two different diseases. Now I want to tell you about an example of where you can mutate different genes that all then produce the same disease. So how can that possibly work? Well, if you think about it, it's pretty simple. The explanation is that these genes which you can mutate, these different genes which you can mutate to give the same disease, must function in a common pathway. So in other words, the genes which you're mutating must all be working cooperatively towards a single goal, or a common goal at least. And a good example of a set of genes that operate in this fashion, and a good example of a disease which arises due to mutations in such different genes, is the disease xeroderma pigmentosa. So xeroderma pigmentosa is generally caused by mutations in any one of 8 different genes that all function in the DNA damage repair pathway. So these are a set of genes which all function cooperatively in order to repair damage to DNA. And predominantly, or a large aspect of that DNA damage repair role, involves repairing damage to DNA which is caused by ultraviolet radiation or exposure to sunlight. Now, you don't have to do a lot of independent critical thinking to try and make the leap between what the consequence would be for somebody who was unable to repair DNA damage caused by UV light. So the major part of your body which is exposed to UV light is your skin. If the DNA is damaged in the skin cells and it is unable to be repaired because there's a mutation in one of the proteins that's cooperatively working towards repairing that damage, then it's likely that the UV is going to damage the DNA and cause skin cancer. And that's what we see in individuals with xeroderma pigmentosa. We see this hypersensitivity to sunlight and this prevalence of skin cancers. This is a particularly nasty case of malignant melanoma that's all over this poor girl's face here. But the take-home message is that it's the same disease, xeroderma pigmentosa, but it could arise due to a mutation in one of any 8 of the genes that are actually functioning in this particular pathway. Genetic heterogeneity, very important you understand that, and you're aware of what the definition of that is and examples associated with it. So in addition to the phenomenon of genetic heterogeneity, there are lots of other examples as well which it's important you're aware of that can affect the phenotype, the disease outcome, in addition to the actual mutation itself. So an interesting one is a phenomenon of penetrance. So the definition of penetrance is the frequency with which a person manifests the gene mutation, I guess I should read, that they possess. So this refers to the fact that not all mutations are actually 100% penetrant. So what I mean by that is that just because you inherit a mutation for a particular gene that is known to cause a disease in one person, there are certain mutations which will not always cause disease in all people. Yeah. So the reasoning for why that is not fully understood, but it's probably down to other genetic factors and environmental factors. So it reflects the fact that there's lots of different variation within our genomes. We're not genetically identical, far from it. We all have subtle differences all across our genome, and those subtle differences combined with differences in environmental factors, whether we smoke, whether we eat poorly, whether we exercise, whether we've, walk up the stairs or take the lift, any set of factors, all operate together to feed into penetrance. And that's how we think it works. That's how we think, why we think some people will be affected by a mutation and some people will not. Now, the degree of penetrance is generally expressed as a percentage. So, for example, if a particular mutation was said to be 90% penetrant, what that would mean is that out of every 10 people who had that mutation, 9 of them would display the phenotype. 1 of them would not. So, a mutation whereby if you have the mutation you always express a phenotype would be 100% penetrant, and a mutation which was very unlikely to cause disease would have a low penetrance, so would have 10% penetrance associated with it. There's a similar phenomenon which is often confused with penetrance, and that's expressivity. So if penetrance is the likelihood that you will display the associated phenotype if you have the mutation, expressivity reflects the fact that there can be variation between phenotype associated with a mutation. So this is a phenomenon whereby the same mutation in the same gene will in one person lead to a very severe disease, but in another person lead to a very mild disease, or perhaps not even a disease at all. And a good example of this is a mutation that causes sickle cell anemia. So all cases of sickle cell anemia are caused by the same mutation, which we'll talk about in a couple of slides' time. And that mutation in some individuals is tolerated incredibly poorly. So poorly that infants will die at a very young age from sickle cell anemia. Whilst other people with exactly the same mutation will often go for decades without being diagnosed with the disorder at all, because the symptoms are so mild. And the explanation for how that can operate are the same as for the explanation for penetrance. It's the fact that people are genetically very different and people are exposed to different aspects of the environment during their childhood, during their life. And that all feeds into and can have an effect on these phenomena of penetrance and expressivity with regards to single gene mutations. Mosaicism is a really interesting factor that can affect single gene mutations as well. And this is defined by the fact that not all cells in the body are genetically identical. Now, it can be confused with X inactivation, which is a different phenomenon. So X inactivation is not a true example of mosaicism. X inactivation occurs in women, so women have 2 copies of the X chromosome. Men only have 1 copy of the X chromosome. But to prevent women producing twice as much as all the genes on the X chromosome as they actually require as an organism, one of the 2 X chromosomes in the cells of women is deactivated and forms what's called a bar body. One of the earliest experiments I did as a geneticist at university was to take a cheek swab from women and from men in the audience and look for the presence of these bar bodies in the nuclei of those cells. So you can often see them as dense spots. So that's X inactivation. Mosaicism is different. Mosaicism is where there's an event very early on in the development of an organism which results in a different genetic makeup for one set of cells than for another. So it can occur at the blastocyst stage, very early on during development of the embryo, during early divisions, prior to the formation of the embryo. Tortoiseshell cats actually arise due to X inactivation, not from true mosaicism. A true example of mosaicism is Klinefelter's syndrome. So we can get a true differentiation between different cells in the body, where some cells will have Klinefelter's syndrome, which is XXY, an extra copy of the X chromosome, and some cells will not. And that comes down to a separation of cell lineage very early on in development. Often diseases which are mosaic are not as severe as the full-blown disease, for the simple reason that often it's only a small proportion of the cells in the body which actually have the genetic mutation present. So phenocopy is another interesting factor you need to bear in mind when thinking about single gene disorders. And phenocopy is described by an environmental effect that mimics a genetic disease. So this is where clinicians can be confused and can misdiagnose an illness as a genetic disease when it's actually being caused by something else. And the best example of this is genetic deafness, which can very easily be caused by deafness which has been induced by the rubella virus during pregnancy. So it's very difficult from the point of view of the clinician to discern the two, to discern whether a child is deaf because they have inherited a mutation in the gene that gives rise to genetic deafness, or they're deaf because a mother contracted a rubella infection during pregnancy, and this rubella virus, which we see here, has led to that infant being born deaf. So that's an example of phenocopy. So there are examples of where genetic conditions can be influenced by environmental conditions. So this is where a mutation can give rise to a disease, but that disease actually itself can be very easily manipulated via changes to the environment. So by altering the environment, you almost eradicate the disease. And the best example of that is a disease called phenylketonuria. So phenylketonuria, if left untreated, it effectively results in a neurological disorder, in a mental subnormality. But it results from the lack of an enzyme called phenylalanine hydroxylase. And what phenylalanine hydroxylase does, it converts phenylalanine in the bloodstream to tyrosine. So if you lack the action of this phenylalanine hydroxylase enzyme, phenylalanine accumulates in the blood of the individual, and if it accumulates to a certain level, then it will start automatically being converted to a compound called phenylpyruvic acid, and it's this phenylpyruvic acid that leads then to damage to the neuronal cells, etc., and disease. Now, it's a good example, phenylketonuria, PKU, of an environmental effect, because it's very easy to actually treat individuals who've got this mutation in the phenylalanine hydroxylase gene by simply putting them on a low phenylalanine diet. So if this is done from very early age, then symptoms don't develop and the person can lead a completely healthy life. So for this reason, PKU was one of the very first genetic disorders that we actually started screening for routinely at birth. So if you're born in the UK, everybody born in the UK has a heel prick taken at birth, and also one taken later on at 28 days by the midwife. And one of the things that the NHS use this heel prick with, this so-called Guthrie spot, is to test for the presence of the PKU mutation. Now, they don't test for the genetic mutation itself, they test biochemically for increased levels of phenylalanine above what would be expected. So high levels of phenylalanine in the blood suggests that the individual might have the phenylketonuria disorder and need to be advised to go on a low phenylalanine diet. So this is a good example of screening. We'll talk about different types of genetic testing that we can do later on in the lecture. So that's an example of an environmental effect where changing the environment, in this case changing the diet of the individual, can have an effect on the manifestation of the actual disease that would arise from the mutation. In this case, untreated, severe mental retardation, simply eating less phenylalanine, completely normal. Anticipation is an interesting phenomenon. So this is the phenomenon, the definition of anticipation, I should say, is where the severity of a disease phenotype appears to increase with each passing generation that inherits that mutated gene. And a good example of anticipation occurs in Fragile X syndrome. So this is again a disorder that leads to mental retardation. It occurs on the X chromosome. And it's caused by one of these trinucleotide repeat expansion mutations that we talked about in the genetic variation lecture. So what we see is, through generations, a steady increase in the size of this trinucleotide repeat expansion. But interestingly, if you have a look at how much this trinucleotide repeat region expands by each generation. What you see is once the expansion reaches a certain size, it will then increase by a much greater size the following generation. So the mutation gets far worse once established as generations progress. A second phenomenon that's interesting is something called genomic imprinting, and this is somewhat complex, but this phenomenon is defined as the expression of a gene depending upon the parent from which that gene was inherited. So this is a concept whereby, depending on whether you got a mutated gene from your mother or your father, can have a direct impact on whether or not you're going to end up having the mutation or not. And effectively, and I don't want to get into too much detail now about this all links into the actual methylation state of that particular gene within your parent's genome. So DNA methylation may be something you've learned about in biological molecules, I'm not sure, but in a nutshell, if you methylate DNA around the site of a gene, it will turn that gene's expression off. So methylation of DNA and chromatin turns off gene expression. Demethylation of DNA and chromatin turns on gene expression 99.9% of the time. And that's what genomic imprinting is about, this methylation of DNA and how it can differ, this pattern, in the mother and in the father. So both of these concepts, anticipation and genomic imprinting, are things that we'll discuss in a lot more detail at level 3 in medical genetics. But it's important you are aware of what they are, and certainly that you know the definition of the two terms for this module. So that's some of the things that can affect different mutations. Then let's consider some of the aspects of nomenclature that surround single gene disorders. So let's start with substitution mutations or missense mutations. So we've talked about these in the last lecture. These are mutations which lead to a change in the amino acid of the encoded protein. So I mentioned earlier that all cases of sickle cell anemia arise due to the same mutation in the beta globin gene. And this mutation, in effect, leads to a glutamic acid being replaced by a valine amino acid. And this occurs at position 6 of the protein. So in other words, if you look at the polypeptide chain that's encoded for by the beta globin gene, the 6th amino acid in that chain, so the 6th amino acid in that chain is normally a glutamic acid. In people with sickle cell anemia, the 6th amino acid is a valine amino acid. So how we write that, how we display that mutation is thus: E6V. So in other words, the mutation is occurring at position 6 of the protein. The original amino acid at that position, or if you like the wild type amino acid, is a glutamic acid, which is an E in the single amino acid nomenclature list. The mutation that's occurred has given rise to an amino acid that's a valine at that position. So E6V. Glutamic acid to valine at position 6 of the protein. So that's how you describe a particular mutation using the correct nomenclature when it's a missense mutation. Nonsense mutations are very similar. So again, we've talked about the fact that Duchenne muscular dystrophy, this muscle wasting disease, often arises due to the presence of nonsense mutations that arise because of frame shift mutations in the gene. So if we have an example where there's a nonsense mutation in the dystrophin protein, we would display it thus. We would say the position of the protein where that mutation is occurring. So this is amino acid 1524. So this is a huge protein, I've already told you, it's a massive gene, dystrophin. And at position 1524, we would normally expect a lysine residue. However, in this particular mutation, the lysine has been converted to a termination codon, a stop codon. So that is represented by the presence of an X. So the X in the single amino acid nomenclature terms refers to a termination or a stop codon. So lysine normally, but at position 1524, that lysine is being mutated to a stop codon, and that will produce a truncated form of the dystrophin protein that would presumably be non-functional. So their missense and nonsense mutations, they have a direct effect on the protein that's been produced by that gene. So we often use mutation nomenclature that corresponds to the actual gene product, the protein itself. You can get mutations which affect just the DNA, and you don't want to go on to then describe the effects on the protein. And these are written out as I've shown here. So familial hypercholesterolemia, FH, basically results in increased levels of fat in the blood, and obviously is associated with heart disease. And there's been numerous studies done on this particular mutation and this particular gene in regards to different mutations present within it. And there was a particular study carried out on a large number of co-sanguineous Pakistani families that had a history of this familial hypercholesterolemia. And what was found is that these families often had the same common insertion mutation present. And this mutation is written out thus. So this is C, which stands for the coding region of the gene. 2416, 2417, INSG. So what this represents is the insertion of a guanine nucleotide between nucleotides 2416 and 2417 in the coding region of the actual FH gene. So that means the actual coding sequence of the DNA. Yeah, not the genomic sequence which will encode the introns and the exons. This is just the coding sequence of the DNA for the FH gene. Deletion mutations. Well, the most famous deletion mutation in a single gene disorder is a delta F508 mutation in cystic fibrosis. So delta F508 represents 80% of all cystic fibrosis mutations that are present in patients from a Western background. And effectively, delta F508 describes a deletion. So the delta symbol, the Greek symbol delta, represents deletion of a phenylalanine amino acid, which is what the F letter stands for, at position 508 in the cystic fibrosis transmembrane protein. So delta, deletion, phenylalanine amino acid, position 508 of the protein. So it's important to understand how you write those different types of mutation. Again, something you might be assessed on later on in the module or in the exam. So if you work in the genetic service in the NHS, how do you detect these single gene disorders then? So I'll have a look at the following website, the website that's linked here. And what you'll see is that there are hundreds and hundreds and hundreds of these genetic disorders now, which clinical geneticists are able to detect by a variety of techniques. So we've gone from 25 years ago really not being able to identify any genetic disorder, single gene disorder in patients, to being able to identify hundreds if not thousands of these things. And in fact, with the advent of PCR and modern sequencing techniques, we can in theory detect every single gene disorder or gene mutation that might be present in an individual. I've already told you in the beginning of the module, it's now feasible to sequence an entire human genome in less than a day for a reasonable amount of cash, £6,000, which isn't a lot of money. Sorry, even less than that, £1,000. So there's a strong argument for, well, why don't we just sequence everybody's genome at birth? But obviously that has ethical implications. We've talked about that as well in the introductory lecture. But certainly PCR and DNA sequencing are used as standard now by clinical geneticists within the NHS to look for mutations in genes, both on a prospective basis, if there's a biochemical result that suggests there might be an issue, and that biochemical result, for example, suggests the person might have cystic fibrosis. One of the things that can be done is to very quickly use PCR and sequencing to scan the cystic fibrosis gene of that person for very common mutations that might be present. And also occasionally other techniques are used in addition to PCR and sequencing to look for errors. So-called protein-based analyses can be used to try and detect disorders such as mutations in the dystrophin gene, which give rise to truncated or shorter versions of the protein, which can be detected via Western blot analysis, which is a technique we use to look at proteins in the laboratory. So that's the technology. What kind of tests are carried out? How is that technology used? Well, we've already touched on newborn screening for PKU. That's a biochemical screen. As I've just alluded to, we do have newborn screening for other diseases, genetic diseases included, particularly cystic fibrosis. So the Trent region, South Yorkshire, Nottingham, they as standard screen for something called hyporeactive trypsin in the blood. In individuals who have cystic fibrosis, the presence of hyperactive trypsin is increased significantly over the normal population, and this is due to damage to the pancreatic tissue in people with cystic fibrosis, which causes leakage of this trypsinogen protein back into the bloodstream. So if at birth, when you have your Guthrie spot done, you've got a very high immunoreactive trypsin level, what will happen then potentially is that your blood on that Guthrie spot will be used in a genetic test to test for cystic fibrosis. Now, in normality, this normally only occurs in families where there's a known record of cystic fibrosis, where people are already known to be carriers for the mutation, normally the delta F508 mutation. But in theory, two people can meet who do not know that they carry a mutation for cystic fibrosis. And remember, 1 in 25 people do carry a mutation for cystic fibrosis. If those two people meet in a nightclub or whatever and get it on and have a kid, there's a chance that kid could just spontaneously develop the disease by virtue of inheriting those 2 mutant chromosomes from each parent. Diagnostic testing, therefore, is linked into that. If you know that you've got a mutation running through your family, you might want to ask for a mutation test to be done. For example, Huntington's disease, if you know that you carry the Huntington's disease allele and you are having a child, it might be that you want to have a diagnostic test done on your unborn child to see whether they've got Huntington's or not, and that might inform a decision to terminate the pregnancy, which I know is a controversial area. I'm not saying I agree with that, but this is a service which can be offered to people who have a history of very, of horrific, uncurable genetic disease in their families. Carrier testing status can be carried out. If you know you're a carrier of delta F508 and you meet somebody, you might want to check that they don't carry a mutation for cystic fibrosis, because if they don't, then it's okay. Even if your kid gets your bad gene, hopefully they'll get a good gene from the father or the mother, and there won't be an issue with regards to inheriting the disorder. Prenatal testing for Down syndrome in women who are a little older and at higher risk is often offered in conjunction with blood tests which are now done, which can detect increased likelihood of Downs, and also translucent nuchal testing, which is done during the actual scan stage of a pregnancy, where they can look at the thickness of the fluid at the back of the neck in the fetus, and that thickness in the back of the neck directly correlates with likelihood of Downs. So if it's above a certain width, then in combination with the results of blood tests, this can inform the decision of whether to go for a prenatal test, amniocentesis, as what's been shown here, which will then confirm whether or not the unborn child has Down syndrome or not. You can have pre-implantation genetic diagnoses, which again might be offered to parents again who know that they have a chance of passing on a very dangerous genetic disorder. You could opt for IVF and make sure that the embryo that gets fertilised doesn't have that particular chromosome present, the mutated chromosome. And also predictive and pre-symptomatic testing. So this will be offered again, to use Huntington's as an example, to an 18-year-old who was completely healthy but who've just found out that their grandparent had died of Huntington's disease. They might want to see whether their parent and ultimately themselves have inherited this mutated gene or not. So just to summarise then what we've talked about how single gene disorders are rare, but they affect a significant number of live births. They generally obey Mendelian patterns of inheritance, so autosomal dominant, autosomal recessive, sex-linked. We've talked at length about the number of different factors which actually alter the disease or the phenotype that a person displays in response to possessing one of these mutations. Talked in a little bit of detail about how you actually go about using the correct nomenclature for these mutations and how these different mutations are tested for within the NHS. So hopefully that's given you a good overview of single gene disorders, how they arise, how the diseases manifest themselves and what can affect that, and how we can test for these disorders in hospitals and in the NHS service. So again, I hope this proves useful during your revision, and look out for more lectures on this topic in this module on my YouTube channel, Born at Bradford. Thanks.